Hey everybody, so I'm finally getting around to making the Meiosis screencast. Sorry about the lateness of it, but you know what, better late than never. Okay, so let's get started. So meiosis is different from mitosis in that it is involved in sexual reproduction. So mitosis basically produces clones of cells. So one cell becomes two identical cells. They're identical to each other and they're identical to the parent cell. Not the case in meiosis. So via some processes that shuffle the genome of the parent, gametes that are produced, so sex cells, end up being genetically different from each other, and that's where we get variation within families. So let's get started on explaining how that works. So, first of all, why meiosis? And in order to address this, we have a karyotype here. So, karyotype is an image that is prepared by arranging pictures of chromosomes from a cell that's about to divide. So not only do they not really line up in rows like this, but you only see the DNA packaged this way when a cell is getting ready to divide. Otherwise, the chromosomes are decondensed and not visible in this manner. So this is a karyotype. This happens to be the karyotype of a genetic male. As you can see, there's a little Y chromosome here. And I like this one because different regions of DNA have been labeled with different colors, so it's a very festive looking karyotype. It's very colorful. So, why meiosis? Well, this is the karyotype from a somatic cell. And somatic cells are the cells of the body, so not sex cells, right? Within each pair of chromosomes, so let's take number 1, just because why not? One chromosome comes from one parent. Let's say this one comes from mom, and the other one comes from the other parent. So let's say that one comes from dad. So within each chromosome pair, there are 2 homologous chromosomes. And homologous means that they have the same genes, but not the same alleles. So let's write that down too. Within each individual chromosome, there are 2 sister chromatids. And within those sister chromatids, each chromatid has the same genes and the same alleles as its sister. So that should give you some context for what you're looking at when you see a karyotype like this. So the DNA is duplicated. That's why the sister chromatids are present. And the chromosomes are homologous. So one from mom, one from dad. And within the homologous chromosomes, one chromosome is going to have the same genes as the other one, but not necessarily the same alleles. So let's move along, shall we? So, just to be fastidious about our definitions, in human beings, nearly all cells, so the exception there, nearly all means not gametes, have paired sets of chromosomes called homologous chromosomes. And we call this condition diploid. So, di means 2 and ploid means number. So, ploidy is the number of chromosomes in each chromosome group. So, humans are diploid. Gametes, sex cells, are haploid so that we can combine them with the cells of another individual. And as we discussed in lab, strawberries are octoploid. So the goal of meiosis is to divide the DNA such that haploid cells result. Because if you're going to combine your gametes with somebody else's to make a new person, you don't want that individual to end up tetraploid with 4 copies of each chromosome. That's not a survivable amount of DNA. That would result in a non-viable individual. Therefore, meiosis is necessary for sexual reproduction. So, 4 haploid cells are produced, and haploid means half the number of a diploid set. So in order to avoid writing haploid and diploid over and over again, we just abbreviate these. So diploid is 2N, and haploid is N. Why N? N just stands for number. So the haploid cells produced are called gametes. The female gametes are eggs and the male gametes are sperm. These are the reproductive cells of human beings and many other organisms. So here's an overview of meiosis. Gametes combined, so one N each. That's not really communicated very well on this slide, so let's add. So sperm has one N, it's haploid. Egg has one N, it too is haploid. When they combine, the N's come together to form two N's, making the zygote, which is the first cell of a new individual, diploid. And hopefully, assuming everything goes well, that fertilized egg will develop into a new organism. But in order for this to happen successfully, gotta make these and gotta make them correctly. So meiosis is how you make these. So this is why you get half your genes from your mom and half from your dad. So this picture is just showing a child's karyotype. So this is offspring. And within the karyotype, it's identifying which chromosome came from each parent. Oops. There we go. So, steps in meiosis. We're going to go through them individually, but it's important to note that one major difference between meiosis and mitosis is that there is one round of chromosome duplication followed by 2 rounds of cell division. Why? That's how you get to haploid. Now, I could, on the exam, theoretically ask you to compare and contrast meiosis and mitosis, and one similarity that they share is in both cases there is one round of chromosome duplication. So chromosomes are duplicated one time, both before mitosis and before meiosis. And then a difference, you could say, would be that in meiosis, there are 2 rounds of cell division rather than just one. So meiosis, because it has 2 rounds of division, has 2 major overarching stages, and those are meiosis 1 and meiosis 2. Each of these go through the same phases, and the reason for this is it's not a naming convention. Meiosis is a modification of mitosis, so asexual or clonal reproduction came first, and sexual reproduction evolved later. So what that means is there are similarities between meiosis and mitosis because meiosis is ultimately coming from mitosis. So same names as mitosis because meiosis evolved from mitosis. And it's always a good idea to think about the evolutionary underpinnings of biological processes like this just so that your knowledge is holistic, right? Okay, so we're going to go through each of these in turn. So in meiosis 1, these things get the designation prophase 1, metaphase 1, anaphase 1, and telophase 1. And in meiosis 2, you just add another, so prophase 2 through telophase 2, etc. So in meiosis 1, homologous chromosomes are separated into 2 separate daughter cells. So each daughter cell ends up with a complete set of duplicated chromosomes, but not a complete set of homologous chromosomes. And that is why, after meiosis 1, the cell is considered haploid. So even though they have a complete set of duplicated chromosomes, that just means that they're duplicated, not that they're homologous. So now we have single N. And then, of course, duplicated means that there's 2 copies, so meiosis 2 is going to separate those 2 copies. So in meiosis II, the sister chromatids of the duplicated chromosomes are separated into separate daughter cells, and each daughter cell has only one copy of each kind of chromosome, and only half of the homologous pair. And half is a little bit loosey-goosey because, of course, chromosomes do perform synapsis and crossing over, so they mix and match their parts, but we'll get to that later. So interphase prior to meiosis looks pretty much just like interphase prior to mitosis. So we have duplicated chromosomes, so this cell would be just coming out of G2, and we have a red chromosome and a blue chromosome. So that tells you they're duplicated. And you can see that they're not condensed in this phase, and the nuclear envelope is still present. Excuse me. I hope you couldn't hear that snort. My bad. I'm still a little bit sick. I'm going to try and keep the nose sounds to a minimum. Okay, so during prophase 1, just as in mitosis, the nuclear membrane breaks down. But as you can see here, instead of having sister chromatids forming, we have homologous chromosomes. And not only that, but they're doing this little dance where they're trading little bits. So they're trading parts of their little legs. So as the nuclear membrane is breaking down, you're also seeing crossing over occur. And you can also see the mitotic spindle beginning to form. So the mitotic spindle, and this should say spindle fibers, the mitotic spindle is the entire structure I've circled, or meiotic spindle in this case. The centrosome is the two centrioles surrounded by some microtubules, but the spindle is everything in the big circle. So prophase and mitosis looks pretty similar. You can see all the same stuff, except for you don't have homologous chromosomes. You just have, you would just have duplicated chromosomes. So those sister chromatids will be pulled apart, not homologous chromosomes being pulled apart. Okay, so during prophase 1, we've got tetrads, which is 1, 2, 3, 4 sister chromatids on 2 homologous chromosomes, and they trade parts, so that's crossing over. Now, I emphasized this at the beginning of the presentation, but it bears repeating. A pair of homologous chromosomes are going to have the same genes. So in this case, they've been reduced to gene A, gene B, and gene C, but they don't necessarily have the same allele. So they could, depending, but in this case, we have somebody who is dominant for all these genes, and this individual has only recessive alleles. But when they cross over, now we have 2 recombinant chromosomes. This one has little a, little b, big C, and this one has big A, big B, little c. So now we have heterozygous chromosomes, and those are recombinant. But there's also some non-recombinant ones. So as a result of crossing over, if you were to split these chromatids apart, which will eventually happen at the end of meiosis, you're going to have gametes of the following genotypes. One is going to be A, B, C. That's this one. One is going to be A, B, little c. One is going to be A, B, C. And one is going to be A, B, C. So crossing over, as you can tell, produces more variation in the resulting gametes. So they're not all the same, which is important. Oopsie. Why no work? Oh, because I have the cursor function on. It is my fault. There we go. Okay, so as you can see in this picture, the metaphase plate looks a little bit differently in metaphase 1 than it does in the metaphase of mitosis, and that's because instead of having sister chromatids aligned at the metaphase plate, we have homologous chromosomes. So this entire structure here is going to be moved to one cell. And now they've crossed over. So another thing to point out is that independent assortment acts here. So independent assortment means that which chromosome is on which side, so these two are homologous, But it doesn't necessarily have to be the case that this one is always on this side and this one is always on this side. If they switched positions, you'd get a different gametic outcome, right? So independent assortment plus crossing over gives you the maximum possible variation, which is the goal here, because of course you want diverse gametes. So here, just to compare and contrast here, here's metaphase and mitosis. So we're going to be pulling these sister chromatids apart, not homologous chromosomes. So, how homologous chromosomes line up determines different genetic combinations in the egg and or sperm. So here's just one more example of independent assortment, and I apologize, the quality of this image is really poor, but as you can see, depending on how the homologous chromosomes line up, you can get different genetic arrangements. So if blue is on blue side and red is on red side, then after metaphase 2, you get big blue, little blue, big blue, little blue, big red, little red, big red, little red. And this is ignoring crossing over. So this is just showing you the possible outcomes from independent assortment. If you reverse a pair of chromosomes, you get big blue, little red, big blue, little red, big red, little blue, big red, little blue. So independent assortment by itself produces quite a bit of possible variation, and if you add crossing over to that, you get a lot more. All right, so during anaphase 1, you see the homologous chromosomes being pulled towards opposite ends of the cells, and it's cute because it looks like they're reaching for each other. They're like, no, don't take me away from my friend. But we have to because we have to make gametes. So, too bad, chromosomes. There is a short interphase, and then the cells move on to prophase 2. So the nuclear envelope briefly reforms. Why? No particular reason, it's just that this is derived from mitosis, so that's why it works that way. And these are considered to be haploid cells at this point because they don't have pairs of homologous chromosomes. But the DNA is still in duplicated form, so we have to cut it in half one more time. So during metaphase 2, this looks a lot more similar to regular mitosis metaphase, right? So we've got sister chromatids aligning themselves at the metaphase plate, but now you can see that they are including recombinant chromosomes. Anaphase 2 pulls sister chromatids away from each other, and the resulting cytokinesis produces, 1, 2, 3, and 4 gametes, and they're all haploid, and they all have different combinations of genetics. So this is the ultimate goal. This is why we do meiosis. So I like this slide because it explicitly compares and contrasts meiosis versus mitosis and shows how they're similar and how they're different. So for that particular potential exam question, you should use this slide to practice your skills. Be able to tell me how they're the same and be able to tell me how they're different. So these are showing you the events in the center of each chromosome during meiosis. So the kinetochores are at the center of the chromatids, and that basically holds them together. So during meiosis 1, you're not breaking the kinetochores. During meiosis 2, you are because you have to pull the chromatids apart from each other. Okay, here's another compare-contrast slide, and this is for those of you who like tables rather than images, because everybody learns a little bit differently. So this shows you events at the top here, and then compares meiosis and mitosis. So it's easy to identify using this which things meiosis and mitosis have in common and which things they do not. So for example, homologous chromosomes line up at metaphase plate. In meiosis, it occurs during metaphase 1. It doesn't occur at all in mitosis. So you might be wondering, yes, they do. They line up at the metaphase plate. How is that true? You have to be careful with your terminology. So this is an example of an exam question that might trip some people up. So homologous chromosomes does not equal sister chromatids. This is a different piece of terminology. So if you can't recognize that those things are different, you might not get that question right. So be very, very careful in how you talk about and think about genetic material in the context of meiosis, because you don't want to accidentally whiff a question just because you weren't paying close enough attention. So, why have meiosis? Well, it generates diversity by ensuring that gametes are genetically different from one another, and that happens via crossing over and via independent assortment. Whoopsie. So variation in offspring makes a population more resilient to environmental changes because remember, some alleles are more advantageous than others, and genetic diversity means that if something comes along that one individual is particularly weak to, it doesn't necessarily mean that an individual who is the sibling of that one is going to also be affected. So from a population biology perspective, it has a positive effect on population survival. So we don't want homogeneous populations because they are easily wiped out by random stochastic events in the environment. So stochasticity means unpredictability, basically. It's a fancy way to say that. So evolution is spurred on by differences among offspring because those offspring are going to compete for resources, and some of them are going to have alleles that give them an advantage, and some are not. So sexual reproduction is a really important piece of evolution for this particular reason. And sexual reproduction occurs because of the process that I just demonstrated to you. So it's important for you guys as biology students to understand that all of the principles of biology that you learn in 160, these are linked. So that's not the case that they're independent of one another. So by now you should be able to link the idea that genetic diversity in populations arises from genetic diversity of combinations of gametes, and that is the substrate upon which evolution and natural selection can act. So by contrast, asexual reproduction is more efficient from one perspective because you don't need another individual. You can just pump out as many other U's as you'd like. However, let's say there's a population of bacteria and they reproduce clonally, so all of them are genetically identical, barring mutation. And in this population of bacteria, they kick around for a while and they're doing pretty well and their colony is growing. But suddenly, they're exposed to an intense ultraviolet light, and there are no individuals in the population that are resistant to ultraviolet light. All of them die. If there was genetic variation in that population, it could have been possible that maybe one of the bacteria was resistant to UV light, and that one survived and went on to reproduce, but that's not the case. So asexual reproduction, while it has certain benefits, leaves populations vulnerable to decimation. And that is the end of the slideshow. So, thank you for your attention. This is actually the last screencast of the term. Hooray! All right, I'm going to post this now, and I will see you guys this coming week. Oops.